Have you ever just stopped and wondered how your body works? How it fights off a cold, builds muscle after a workout, or even just digests your food? Well, the answer, for the most part, is all about millions of tiny, microscopic machines called proteins. But here's the wild part. These incredibly complex machines all start out as nothing more than a simple string. So how in the world do they get the specific shapes they need to do their jobs? All right, let's unfold this mystery together.
So first up, we've got to appreciate just what these proteins are. I want you to think of them as the body's little engines, or maybe sophisticated nanomachines that are constantly buzzing around inside every single one of your cells, performing countless jobs. And when I say workhorses, I really mean it. Proteins are not one-trick ponies. They are, without a doubt, the most complex and functionally clever molecules we know of. They've been fine-tuned over billions of years of evolution to handle an absolutely incredible variety of tasks.
I mean, the sheer range of what they do is just astonishing. They're the structural scaffolding that gives our cells their shape. They're the enzymes that speed up chemical reactions by like 1,000,000 times. They're the delivery trucks, like hemoglobin, carrying oxygen through our blood. They're also messengers, sending signals between cells, and there are bodyguards defending us as antibodies. They even help our muscles contract. It's just, wow.
So this brings us to the big puzzle, right? How can all of these wildly different and complex machines, each with a super specific job, be built from what is essentially just a one-dimensional string of chemical building blocks?
This is really the heart of the matter. How does a simple linear chain, kind of like a string of beads, just spontaneously fold itself into a very precise, stable, three-dimensional structure? The answer is one of the most elegant stories in all of biology.
The whole secret to how that string becomes a specific shape, well, it begins with the code that's already embedded right there within it. Those beads on the necklace, they're called amino acids, and the specific order they're in, that's everything.
This sequence has a name. It's called the primary structure. And here's the most important takeaway. All the information needed to create that final, complex 3D shape of a protein is contained right here, in this unique order of amino acids. The sequence is the blueprint.
And what's truly remarkable is that this incredible diversity of protein shapes and all their different functions comes from an alphabet of just 20 different types of amino acids. That's it! And each one of those 20 has its own distinct chemical personality, and that is the key.
So the main thing that kicks off this whole folding process is how these amino acids react to water, which, fills our cells. Some of them are hydrophobic. They're basically water-fearing. Think of them like oil in water. They desperately want to get away from the water, so they bury themselves deep inside the core of the protein. But others are hydrophilic. They're water-loving, and they're perfectly happy to hang out on the outside. This really simple tendency is the main engine that gets the whole protein to start folding up.
Now, this folding doesn't just happen all at once in a big messy clump. Nope, it's a very orderly process that scientists break down into 4 distinct levels. So let's build a protein together, step by step.
Okay, let's watch how this happens. We start, of course, with the primary structure. That's our unique sequence of amino acids. Then, little sections of that chain start to form these regular local patterns. That's the secondary structure. Think of elegant coils called alpha helices, or these rigid folded sections called beta sheets, almost like a paper fan. Next up, the whole thing, coils, sheets and all, collapses into its final compact 3D shape. This is the tertiary structure, and it's where that powerful hydrophobic effect really comes in to lock things into place. And for some proteins, the journey's still not over. They reach a quaternary structure, which is where multiple folded-up chains come together to form one giant functional machine, like hemoglobin.
So, okay, we've gone from a simple string to a complex shape. But why does this one specific final shape matter so much? Well, the answer takes us right back to why proteins are the workhorses of our cells in the first place.
There's this quote from a classic biology textbook that just nails it. Basically, a machine with a wobbly, unpredictable shape is, well, it's useless. For a protein to do its job over and over again, reliably, it has to fold into one single stable shape. That precise shape creates all the little pockets and surfaces it needs to grab onto other molecules and get to work.
So here's the bottom line. The linear sequence of amino acids determines the three-dimensional structure. And that specific structure, in turn, determines the protein's biological function. It's a direct chain of command, from information to shape to action.
And this is why protein folding is so incredibly critical for our health.
A single mistake, just one wrong bead in a chain of thousands, can cause the whole protein to fold incorrectly. This wrong shape means it can't do its job, and that can lead to devastating diseases like cystic fibrosis or sickle cell anemia, and it even plays a role in conditions like Alzheimer's. When it comes to proteins, shape is absolutely everything.
And that brings us to today and to the future. For decades, being able to predict that final 3D shape just from the amino acid sequence was one of the biggest challenges in biology. But now, with huge advances in AI and computing, we're getting ridiculously good at it. And this raises an amazing final question. If we can reliably predict how any protein will fold, what kinds of problems could we solve? Could we design new medicines that fit perfectly into a disease-causing protein and just turn it off? Could we create brand new enzymes to break down plastic waste? Understanding this fundamental mystery of life is unlocking a future where we might be able to design biology itself.